So thank you everybody for coming. And as Nomi and Peter said, I'm going to be giving most of this presentation as if it's 25 years from now, which is when I'll be roughly 65 and looking for a new career beyond what I've been doing for the last quarter century. I thought it would be nice to lead with this excellent quote. It's attributed to either Niels Bohr or Yogi Berra, which says, "It's hard to make predictions, especially about the future." So take or leave their next 30-odd slides. All right, so I'm going to give a history of bioinformatics over the last quarter century. And I think it's rare that you get a chance to sit back and reminisce about your career in a science for a whole hour. So you'll have to put up with me for this for a good portion of this hour. And one of the reasons I was invited is because of the Brain 2050 initiative, which I'll talk about towards the end of my talk, which is a large newly funded investment in neuroscience. And I think that there are some lessons from the last quarter century of bioinformatics and biology that could be usefully applied to the future studies in neuroscience. I'd also like to point out, for those of you that are relatively new to this field, that about 20 years ago, bioinformatics and systems biology really disappeared as far as terms went, because they simply started to be referred to as biology. The sort of artificial distinction between the computational and data analysis sides and the modeling sides of biology with experimental biology disappeared, along with many of the experimentalists, which I'll talk about in a little bit. So the 20-teens and onwards really saw a lot of change. And I'm going to focus on three sort of semi-apocalyptic contributing factors. One was the too much data apocalypse situation. One was the reproducibility crisis. And the third is computation in biology more generally, computational training in biology. So I think many of you are familiar with the fact that at one point there was so much data that and so little computational infrastructure that we really had a tough time dealing with the data. We still have a tough time dealing with the data, but we're much better prepared now. And again, that's what I'll be talking about a little bit. So this data apocalypse really came from a collection of several different things. One was the vast increase in omics data generation capacity. So this started with genome sequencing and transcriptomics and metagenomics, but grew into metabolomics and proteomics to the point where essentially anything that you could get a sample of, you could sequence, you could characterize at a protein level, you could characterize at a metabolome level, and that became connected with automated sensor data. So we were sending underwater drones to cover much of the ocean and sample automatically. We were sinking sequencers to the bottom of the Mariana Trench to see what was down there and sample on a continuing basis. We were sequencing the air in really all the major cities. And essentially, between the data generation and the data sharing between different projects, biology became a data-intensive science. The first set of problems, the volume, velocity, and variety problems that was sort of in vogue, the three V's, is sort of a general problem that a lot of science was suffering from at the time. So physics and astronomy and chemistry all had lots of fast-moving data of a wide variety of formats. But the big problem in biology was that biology had spent the 40 years prior to this optimizing themselves for hypothesis-driven investigation, not data exploration, which meant that when this data came along, all of the students and all of the faculty and all of the funding agencies were completely misaligned in terms of what their focus was. Not to say that that the new focus doesn't include hypothesis-driven investigation, but that the data exploration side was very weak. And so, unfortunately, and this is when I was starting my career, this was a really big problem for people that were starting their own labs. There were long arguments over whether or not we should be doing more data exploration or more hypothesis-driven investigation. And unfortunately, the people who controlled the funding, which tended to be the older, more senior faculty who had made their reputations doing hypothesis-driven investigation, they tended to win, which represented a real challenge for the future of the field. The other thing to point out is that at the time, the Moore's law had continued to increase the amount of computing that was easy, but a lot of the I/O challenges were completely unaddressed, and that continued through the early 2020s. So compute was cheap, but not throughput. And when your compute depends on getting the data to compute upon, this means that essentially you're wasting most of your compute power and most of your software. Okay, so the reproducibility crisis. In 2015, we started to systematically examine papers to see whether or not they could be reproduced in their particulars. And what we found was that if you were fairly picky, about half or more of the papers simply couldn't be reproduced. And by picky, I mean there were parameters missing, there were details of the analysis missing, there were statistical cutoffs that weren't specified, there were versions of the software or versions of the data or data cleaning steps or plotting components that were simply ignored in the publication and that couldn't be recovered without actually going and talking to the lab. In some cases, they couldn't be recovered even if you did go talk to the lab that had done the research, of course. But the goal of having a paper and being able to understand exactly what had been done to go from raw experimental work all the way through to final data analysis was essentially, that goal was unreachable, was unattainable. And what you can see is that as we systematically ramped up our investigations of reproducibility in response to a number of very high-profile replication failures in the 2015, 2016, and before era, there was a large number of failures that slowly started to decrease around 2022, which I'll talk about in a few slides. So something that might be a little bit hard to understand from the vantage point of 2039 is why was there this reproducibility crisis? The interesting thing is it wasn't particularly surprising to many people at the time. It's a well-known fact among biotech, it was a well-known fact among biotech at the time that most published experiments were essentially useless for building off of for therapeutics. That is, you would go, you would read a paper, you'd say, wow, that was a really interesting experiment. There'd be some mouse line, you'd try and get the mouse line. If you could get the mouse line, which was always a challenge, you would find that details of the experiment, details of the cell culture, details of the perturbations were simply impossible to recover. And this led to a lot of wasted biotech effort. And the reason behind all of this, of course, was that the career incentives simply weren't there. So neither the career incentives nor the funding, which basically amount to the same thing, were there. And in fact, the contrary was in place, which is that if you could publish a splashy result, if you could get your result past reviewers, that was what made your career. You didn't have to have a reputation for good reproducibility. You didn't have to have a reputation for detail-oriented work. You just had to pass yourself off as if you were doing detail-oriented work. This slowly started to change later in the decade as the public caught on, right? If the public sees billions and billions of dollars going to, for example, cancer research, and cancer biotech companies say, hey, all of this is wasted money, then funding starts to decline and bad things happen. And that is indeed exactly what happened here. It's a little bit hard to believe now, but back then, if you claimed to have seen something, that was sufficient for a high-profile publication. There was no independent replication. And the utter disaster that this led to in terms of the funding crunch for experimental biology was only addressed by a rather draconian two-lab rule where 2 independent labs had to reproduce a result before it could be published. This was really helped by members of this community where papers started being rejected when they didn't have a replication report with them. And of course, once papers were rejected without comment for no independent replication, the funding shift soon followed because nobody could get publications, so nobody could get grants. Okay, so the third component of the apocalypse was of the sort of the mess in the early 20-teens was that it was computing and data in biology. So of the sciences, biology had always really been the weakest in terms of computing education. And so when I was starting, there was this essentially a complete disaster where I would regularly talk to graduate students and labs that were generating data sets or trying to make use of data sets that they couldn't analyze, they couldn't understand, and in many cases for which no approaches existed. They were generating data sets and saying, well, you know, I'm sure if we generate the data, we'll be able to analyze it. And of course, that was completely and utterly false. And this resulted in a number of problems for graduate students and early career faculty at the time, and led to a shift in emphasis that, again, I'll talk about in a bit. So one particular thing I'd like to focus on, and we're past this in bioinformatics and biology, and I'll talk about it a little bit in the context of the Brain 2050 initiative. There was an immense investment in the late 20-teens when a number of senior biology professors said, look, we just want push-button data analysis. We want to generate the data, we want to feed it into a computer, we want our results, and we'd like a science paper, please, at the end. This worked pretty well outside of research, especially in clinical areas where the problems were well understood and the data analysis pipelines were in place. It turns out that in research, most data analysis can't be placed in a black box and well understood. And these easy-to-use tools really embodied so many assumptions that a lot of the results that came out of them were simply invalid. And that didn't come to light until we started doing the replication and people started understanding what the details of the analyses were. So this was an interesting sequence. So one of the things that infuriated me when I was a young faculty member was this concept of turning the crank. I don't know how many of the older members of the audience remember this, but it was common to go to graduate committee meetings or grant review panels where people would say, gosh, you know, they're generating a bunch of data, the tools already exist, surely you just need somebody to push the right buttons and that can't be that hard, right? Right. Of course, to sort of refer to what I said in the previous slide, it turns out that the expertise and training needed to build the tools or construct the pipelines or even use the pipelines was actually quite significant. And so this meant that you had a bunch of poorly trained graduate students pushing buttons that they didn't understand. This led inevitably to a very unfortunate series of events in the bioinformatics community at the time. The concept of bioinformatics sweatshops. They didn't look like this, but essentially that's what they were. There were a number of graduate students and low-paid employees that were devoted to service bioinformatics. They were immured in the corners of labs where they couldn't drink coffee, which also turned out to be a really bad idea. There was no career path for them beyond their PhD. There was no significant authorship granted to them because after all, they were just pushing buttons and turning the crank. But it turned out because it was necessary to have these people around in order for big labs to make progress, in quotes, they existed for about a decade. Okay, so some of you may remember when things came to a head. So essentially, there was a lot of effort spent on figuring this out. The well-trained students didn't really have a problem. They left for data science on the West Coast. More and more papers were being written by people who didn't understand the computing as a result, right? Students would get their master's degree or their PhD and they'd go on and they'd sit in the corner of some bigger lab and they'd be stuck on some project they didn't really understand, using tools that they didn't understand to generate results that were probably wrong. A lot of those papers were then rejected, but of course the supply of reviewers also ran out because we simply weren't recruiting these people into the ranks of faculty. And then, well, California. So we may never know exactly what happened given the interdiction zone and the lingering contamination, but it turns out that while the data was replicated globally, the expertise was primarily based in the Bay Area. And this meant that we lost in one fell swoop, we lost about 80% of the data scientists in the world. This really exacerbated the review situation. I mean, it was a tragedy, obviously, but it's far enough in the past that you all know that history. But it really exacerbated the review situation. So at this point, the remaining bioinformaticians and data scientists essentially revolted and created a union. This was not an official union because by then the Republicans had banned that. But essentially, we came up with 3 rules. All of the data and source code must be provided for any paper, period, before we would even review it. Full methods sections and references needed to be included in the primary paper review. And no unpublished methods could be used in data analysis. So the interesting thing about that was if you did a bunch of new bioinformatics, a bunch of new algorithms and tool generation, and you included that in your exciting new biological discovery paper, you had to have another paper on the methods. And it had to be made available at the same time and be subjected to the same peer review. Because ultimately, what everybody had known for a long time, but we really only instantiated as a rule in the community was that the quality of the underlying, of the overall work depends critically on the methods, both experimental and computational. And so what this led to, of course, was a lot of preprints, which meant that the only companies that really complained were companies like Microsoft Elsevier, because it led to a vast increase in preprints. Another thing that I really wanted to highlight is the concept of replication parties. So the community practice emerged around replication in the late 20-teens and early 2020s as the depth of the reproducibility crisis became clear. This is something that was primarily computational at the time, but it led to a really wonderful increase in the number of people that understood what was required for computational reproducibility. Essentially, the explosion of open peer review meant that when papers were published, everybody could read the reviews of the paper. This actually was put in, this was called the arsenic life rule. And when the peer reviews were bad, these papers were then nominated for a replication study. And these were internet scale distributed replication parties where independent groups would attempt to reproduce various aspects of the paper. And a number of journals emerged to publish those replication attempts. And a number of the mid-career faculty in this room actually, I know, enjoy their careers due in large part to these publications, because they highlighted so many different problems with the bioinformatics approaches that were being used at the time. They led directly to the development of many new methods and techniques. But in general, what this meant was that going forward, graduate students that participated in this not only networked, not only learned how to do things, but just published better papers down the road. So I don't think that the bioinformatics, the computational community, can lay claim to the entire renaissance in biology. There were a number of things that contributed, starting in 2020, to just a complete and utter shift in the way we thought about biology. One was the biomedical enterprise rediscovering that basic biology existed. The second was the rise and ultimately the triumph of open science. Third was a transition to network science, which actually accompanied some pretty catastrophic career transitions. And then the fourth was actually the realization that we needed to invest massively in people to make progress. So for rediscovering basic biology, it turns out that a really underappreciated facet of the rise in data generation was that all of these organisms that couldn't be studied in the 90s, in the 1980s and 1990s, could now all of a sudden be studied in great detail at an omic level, at a genomic, transcriptomic, metabolomic, proteomic, and ultimately environmental level. So we'd always known that mouse, for example, was a fairly poor model for biomedical research. And a number of veterinary and agricultural animals proved to be better models for various kinds of genetic diseases. Ecology and evolution had a deep history of theoretical and empirical work that could be used to help understand human genetics. All of the microbiome work finally started to get integrated into the medical establishment because of things like the built environment and disease reservoirs, where in order to understand the causes of even early childhood diseases and things like autism, you had to bring in the microbial component. And then, of course, cheap sequencing enabled a vast array of these studies. Open science triumphed. So the second component of the Renaissance was that open science really became sort of de facto. So I would say that the computational community really knew this by 2016, but of course it took a few years for the rest of biology to come around. And the story is kind of interesting because I think it can provide direction for future efforts. Biotech basically noticed that all of the experimental money that the NIH was spending on biomedical research was going down the drain. Because either the experiments were badly done or the analysis was wrong, and the raw data was never available until it was published in a high-profile journal. And this meant that the raw data was never available once the reviewers got a hold of it. A more general funding crunch tightened the screws further, and hypothesis-driven labs basically couldn't compete. The labs that were focused on single gene studies, the only things that were really tractable with pure hypothesis-driven research, simply couldn't compete anymore with the rest of science. And so this led to a shift where hypothesis-driven lab science joined with discovery, so that when you were trying to decide what experiments to do, you would look at the panoply of available data, build your hypotheses based on that, look for other data to cross-validate with that, and only when you were pretty sure that the hypothesis had no data that contradicted it, did you go in and do the experiments. And so this led to a blossoming of data-driven modeling and model-driven data interpretation. Then there was the transition to networked science. This took a surprisingly long time, but it helped that the universities collapsed. What this meant was in effect that all of the senior faculty retired. This was a challenge from the history of science perspective. There was a massive brain drain at the time. Many theories and concepts had to be rediscovered from scratch by the Young Turks, but it also enabled a massive increase in creativity at the research level, because there were no wise gray-haired people in the meeting saying that can't possibly be true. And there's a maxim, right, that's still in force today, that if a senior scientist says that's probably possible, they're probably right. But if a senior scientist says there's no way that could possibly work, they're almost certainly wrong. And so by getting rid of that negative influence at the tenure level, at the promotion level, at the granting and review level, it led to a massive increase in creativity. And when you added in the sort of high quality review and replication components, science didn't actually suffer that much. This led to a real shakeup in collaboration tools, data sharing, and really the rise of distributed team science in a way that we hadn't seen before. One thing that I wanted to call out specifically was this walled garden model. And this was, it's a model that's now obsolete because we're sharing, as everybody knows, we're sharing all data all the time now, but it was quite effective back then in driving the transition to open data and open source and open science. Data collection was essentially done by small consortia, and the data within that consortium, the data was made available to everybody in the consortium, but then published in lockstep. It's actually followed from a model that was popular in, for example, ocean expeditions, where the upfront investment to charter the boat for the cruise was so high that you had to get a consortium together in the first place. But Sage Bionetworks made this work in the context of biomedical research in the early 20-teens. And this then led to people understanding that there wasn't actually that much to fear from publishing your data first and then working on analyses later. Finally, I should say, with the pioneering appointment of Phil Bourne to the Associate Director of Data Science at the NIH finally realized that training was important. They'd always had some training grants, but they tended to be for doctors, and they tended to be for only at the graduate and postdoctoral level. So one particular thing that I'd like to highlight, and again, most of you in this room have never seen these words because they're just part of training now, but there were 3 different efforts that we put a lot of time and effort into: software carpentry, data carpentry, and model carpentry. Those are now simply folded into scientific training. At the time, it was actually pretty novel, the idea that we would actually train people in how to do things properly computationally, which I think shows how backwards the whole field of biology was in 2014. There's still lots of problems. I don't want to pretend that we've solved everything, but I will say that those problems are things that we can at least appreciate and work on. So what do most genes do? We still don't know. Functional annotations are very poor for most organisms. Some approaches in biogeochemistry and synthetic biology are really pushing on this. Career paths for experimental biologists are still surprisingly uncertain. We have the opposite. We have a new problem with what's being called GLAM data. Now that data sets can make a career, there are people that specialize in publishing high-profile data sets without doing any analysis on them. And this turns out to be just as lopsided as publishing high-profile analyses without making the data available. So, we make our own problems in science. As the field swings from side to side and emphasis to emphasis, we ignore things that we shouldn't ignore. And the rise of data science and hypothesis-free investigation has led to actually us ignoring hypothesis-driven investigation, perhaps too much. I would say cancer is pretty much cured except for a few edge cases, but most of the complex diseases remain poorly understood. In some cases we have therapeutic remedies, but it turns out anything that involves more than a couple genes is still impossible to understand. It's simply a network effect, which we'll talk a little bit about later on. So, all right. So I was invited here to speak in part because of my quarter-century history in bioinformatics, my 30-year history in bioinformatics, but also because there's now a newly proposed 10-year large multi-trillion-dollar project to understand the brain by 2050. And this actually builds off of, this is a new initiative that you may have seen announced by President Rao. And she's based this initiative off of an initiative that was just being announced a quarter century ago, the BRAIN initiative by President Obama. This one is focusing on neurodegenerative diseases, regeneration, and sort of generating a mechanistic understanding of intelligence. And I think that we can look back over the last quarter century of biology and computational biology and bioinformatics and maybe make some suggestions for how the new neuroscience programs can systematically invest to make things successful. Okay, right. This slide would have been current 100 years ago. It's still current today for reasons that I don't think anybody in this room can understand, but I want to mention it. Correlation's not causation, right? The original MIND project 25 years ago failed for this reason. They essentially said, hey, if we record all the neurons all the time, surely we can understand the brain. And it turns out that the more data you generate, the more things you record, the more things you observe, the harder it is to extract any sort of meaning from that data. The more time and investment you have to put into tools and analysis and models. And so in this case, we could record everything, but we still had no understanding of causation. We still had no understanding of the network effect. And so the funding for this lapsed, and the whole project collapsed by 2020. I think it's worth pointing out that computational modeling is critical. And if there's one regret that I have of the last 25 years in bioinformatics, is that bioinformatics and biology did not embrace modeling. They did not embrace data-driven modeling to the extent that the neuroscience community essentially already has. We need to do a better job in all communities of generating models that embody hypotheses that we can then test on the data, both by generating fake data sets, seeing how our tools perform on them, and correlating the data sets that we do generate with the data sets that have been generated under certain hypotheses. And this concept of holistic multidisciplinary research is not a new one by any means, but it is an overall ignored one. And again, I would say the brain community has always been better off here because we were already aware that the brain was tremendously complex. Whereas for some reason, the early yeast and E. coli geneticists thought that things were simple, and that transmitted itself to the biology community in the late 90s. So here's a funny call for me. I think we need to focus less on reproducibility. It turns out that being strict about independent replication strangles forward progress. The 2-lab rule means that often it takes 2 to 3 times as long to publish a study as it used to. This is great for the solidity of science, and it's completely understandable given the disasters of the past in terms of both funding and public opinion, but it's also incredibly slow. So can we compromise? And here I'd like to bring in a very successful concept from software engineering, the concept of technical debt. Here we're calling it replication debt. And the idea is essentially that so in software engineering, there's this idea that an early focus on maintenance and cleaning up old crap, so to speak, can actually lead to an acceleration down the road of progress. Of course, the problem is that research is always in this phase down here where we're generating new data that we don't entirely understand, and as soon as we understand it, we move on to generating new data. The idea of replication debt, much like technical debt, is you do rapid iterations of analysis and then go back and move to a second stage where you go back and re-examine the quality of your analysis and re-examine the replicability of your analysis once you're sure that it matters. So the concept of replication debt is essentially that after an initial exploratory phase, you do allow semi-independent publications, semi-independent replications, not completely independent replications. So someone might go visit the lab that originally generated the data and make sure that they can work through the protocol in that other person's lab. This deals with issues like sources of enzymes and bespoke computational infrastructure and lab water, which turns out to be a big issue. And then you follow that with an articulation of the protocols and independent replication once you're sure it matters. And the most important bit is that this debt has to be publicly acknowledged. So when you publish these papers, you have to say, this hasn't been perfectly replicated, so you might not want to trust it. And this actually is then a key for funding agencies to say, gosh, a number of people want to build off of this result, but it's not yet solid. We should probably make it solid. We should probably fund those proposals that want to independently replicate it. And I think that this is a better path forward than strictly requiring independent replication. So something else that is going to sound a little bit strange because it's obvious is Brain 2050 should really invest in infrastructure for collaboration and data sharing. This is a given, but I think the existing tools really only support only support data science sharing. They don't really drive that sharing. And I think that a push for collaborative process from the outset would be great. And here I'm going to steal a concept that Phil Bourne published about 25 years ago that essentially when you gather and deposit data, this can be automatically compared against other data sets, and people that might be interested in your data set can be notified about the things that you're doing, the models that you're generating, the data analyses that you're doing, the tools that you're building. And I think that this could help drive more collaboration between labs that may not even know of one another. So we still have the community silo problem, of course. Okay, tool up. Again, obvious. You need to build a robust and competitive software ecosystem. You need to have that ecosystem there for innovation and creativity. All of these tools, of course, need to be available, open, reusable, and remixable. But we need to invest more time and effort into understanding why different tools do the different things that they do. So in the computer science community, there's been a big emphasis on benchmarks. Benchmarks are a really bad idea because, in general, because you can always spend a lot of time doing engineering optimization to eke out that extra 10%, but rarely will you understand how your algorithm differs from a radically different algorithm that may reproduce similar but not overlapping results. And that's what we're going for in research. There's been a great tradition of that's developed over the last 20 years of building commercial software based on high quality scientific research software. So the idea is companies that want to build software and sell it to scientists build on top of popular protocols that are well understood, which in turn emerge from research development within labs. So rather than companies developing their own algorithms, which nobody understands, keeping them under keeping them under lock and key, and then touting them as being improvements to standard algorithms. The standard algorithms emerge from the research community, are integrated into popular protocols, and only then do companies build easy-to-use interfaces around them. I think this is actually a great alternative to the approach. This is a great addition to the approach that I discussed earlier, where the black box nature of data analysis can't be, you can't black box most data analysis. Here you start with sort of white boxes, open boxes, and then encapsulate them in GUIs and user interfaces when the time comes, when they're well understood. Something else that I think we continually rediscover is that we should be investing in training as a first-class research citizen. So yesterday's K-12 students are going to be the research scientists of tomorrow. We need to start here, train them as undergraduates, and then even though the term graduate student is sort of a misnomer given the rise, the failure of most universities and the rise of research institutes, people still do receive PhDs. And we need to have them trained before they come to start doing research. One interesting thing about this is that the massive investment of the NIH at the K-12 level has led to an 80% acceptance of evolution as a major fact in biology among the 30 and under set. And now we just need to wait for that to take over. So there's a lot of good benefits to this. I think at this point I'll just say, so that's my set of advice. I think it's worth saying that single molecule full genome sequences, when they emerged in the late 20-teens, didn't provide understanding. Reductionist studies of gene function didn't provide understanding. High resolution ensemble neuronal sampling won't provide understanding. Our main obstacle in understanding things like aging has been that these things are system effects. They require modeling, data analysis, and perturbation experiments. And this is hard to coordinate. I would like to also point out that for neuronal studies, we actually have a big problem because it turns out most of the neuronal perturbations are unethical. And so we have to rely on models and data generation to a much greater extent than the organismal biology community did. Okay, so some concluding thoughts. There are many things the Brain 2050 field can do to invest in its own future and accelerate progress. Every field fails to learn from the mistakes of the fields before it. Perhaps maybe we can do better moving forward. I would say that many of the mistakes that biology made in the last quarter century were things that physicists and mathematicians and chemists already knew and simply weren't being translated into the biology community. All right. Okay, so that's pretty much, I mean, I could start going further off the rails, but I don't have more to say about my future predictions. I do have a couple more slides. When I came up with this talk, which was not a sane idea, I thought I wanted to use this talk as a way to highlight a set of future issues that I think, to sort of explore in a science fiction sense, the set of future issues that I think face us today. But it turns out nobody can predict the future, right? So there's this great book, and I remember when this book came out, I was just starting graduate school in '97. And John Horgan published this book, The End of Science. Now, I don't know how many of you read John Horgan. He's like the most massive troll in existence. He essentially comes out with these strong statements like, I forget what, the science is coming to an end was one of them in 1997. Another one was I think the internet hasn't been useful or something, or something. And you're like, what are you, and you know, I think I'm hoping that he's putting these things out there to discuss. But there was a great comment. Among the pessimists, molecular biologist Gunter Stent suggests that science is reaching a point of incremental diminishing returns as it comes up against the limits of knowledge. This was in 1997. Could I just get a quick show of hands for those of you who believe this quote? No, okay, right. So, admittedly, it's only been 15 years. Maybe tomorrow we'll stop finding new things. So, this comes via a good friend of mine, Eric Schwartz, who's currently at Cornell. And he and I are both fans of Robert Heinlein's writing. Now, Robert Heinlein, for those of you that aren't science fiction aficionados, was one of the big three, along with Arthur Clarke and Isaac Asimov, one of the big three science fiction writers of the early 20th century. He had a lot of bad ideas in his books. We'll just leave it there. But he also thought quite deeply about the future. And so, in 1950, he published this monograph, Where To? And he put out 4 curves of predicted human progress. So the solid curve here, you know, this is hand wavy, right? The solid curve represents anything you care to mention. So let's go with the cost of sequencing, the decreasing cost of sequencing, or the increasing amount of data available. And you can call this the curve of human achievement. And yet, people persist in predicting, in under-predicting the future. So I guess, I don't know who the patent office official was of 100 years back, I guess 1850, given that he was writing this in 1950. He quit his job because everything had already been invented, right? This was actually a Victorian sensibility, right? That things had reached not just a local optimum, but a global optimum. I guess if you're a rich English person in the 1800s, then maybe that was a global optimum for you. But they tended to underpredict. Those who are a little more optimistic expect us to reach a point of diminishing returns. And then the really daring minds, and I don't know that I classify myself among them, but at least the people that sit and write science fiction and try and predict the future, predict that we'll continue sort of in a predictable way, right? A tangent to this curve of human progress. And this is all crazy, because if there's anything we can say looking back over the last 15 years, it's that there will be a tremendous amount of change that cannot be predicted at all. And so I have no idea what's going to happen in the next 10 years, much less the next 25 years. I think that if I have to give you a message, and this is a keynote talk, so I'm not supposed to talk about anything solid, right? We're supposed to say something inspiring and empty. We should invest in people and process. We have to figure out what the right process is, and then provide career incentives for people to do things that way. And I'm really looking forward to Phil Bourne's talk, because he's the guy with all the money. You know, he's going to be talking, I think, about career incentives tomorrow. This community should be leading the way. I think Nomi's introduction was perfect. Open source is a done deal, right? We're going to win. We won that. Open science, less so. I think we're going to win. But we need to set the terms of what the community should be doing. We should be thinking through these things. We should be trying things. Everything that I do, Peter said some very nice things about the open science efforts in my lab. It's a little hard to describe now that things seem to have worked out pretty well. Like, I've gotten some grants, which wasn't sure 5 years ago. I published some papers, which wasn't sure 5 years ago. People are using our source code, which wasn't sure 5 years ago. Now that seems to have been working out, it's worth looking back and going, I had no idea what I was doing, right? I mean, it's a little hard. People always like to tell a story about their backstory, like, yeah, I figured it out from day one, right? Total nonsense. I have no idea what I'm doing, but I'm trying new things. And if that's what I think we need to do as a community, we need to try new things, develop new process, figure out what makes sense and what doesn't make sense, and then do the stuff that makes sense and convince others that it makes sense simply by doing it. On a slightly more sobering note, economics do matter. So this is a 50 million mark note from Weimar Germany in 1923. Inflation, for those of you that don't get that message, right? And I think it's really entertaining to look at the USGAO predictions, right? So this is right now, and then these are their projections. And the best description of this, where you have Medicare, Medicaid, Social Security, and defense taking up the lion's share of the budget, is that the US is becoming an incredibly well-armed pension fund. And the reason I want to show this is because all of science is in here, right? All of scientific funding is in here. And the non-interest spending is decreasing and decreasing and decreasing and decreasing relative to the available money. And this is grim. I have another talk that I gave. I have another section to talks that I give called Planning for Poverty. You should be planning for decreased investment in everything. How can you continue to make progress with less and less money? So why am I saying this? Because I think that one of the edges that the open source and open science community has is that the data sharing and openness maximizes the return on investment for funding agencies and for the public. And that's ultimately what matters. If we can convince people that the money they're spending on research is actually doing something useful. We've been spectacularly unsuccessful at this, by the way, over the last decade or so. If we can convince them, then there's at least the possibility that we'll get maybe neutral budgets rather than negative ones. But in order to do this, we also have to figure out how to align the career and funding incentives, because it turns out that virtually no sane assistant professor will invest in these data sharing and openness heavily without some assurance that they won't lose their job in 2 years. We are currently doing a horrible job of this, and as I said, I'm looking forward to Phil Bourne's talk. So I have a couple of people I want to thank. I actually had a long conversation with Phil Bourne before I was invited to give the keynote, and some of this comes from that discussion. Eric Schwartz contributed a bunch of the more sobering concepts. Katrin and Jeffrey Campbell, both at OSU, talked with me quite a bit during a visit there about this, because I was wondering, what the hell am I going to talk about? I think I'd already given Nomi the title, so I knew it was going to be something kind of crazy, but I wasn't sure what. All of this will be or is already posted online. In fact, let me just. Yeah, but I also have my talk notes are just going up now. I took negative inspiration, and I mean this in a good way, from the Next 10 Years of Quantitative Biology talk by Mike Schatz. This talk is fantastic. Email me if you can't find it online. But I treated everything he said in that talk as completely obvious for the purposes of this talk. I don't know if that makes sense, right? He made a bunch of predictions about what sequencing was going to do over the next 10 years, and I said, if he can, and I believed it all, and it was great, if he can predict that 10 years out, I shouldn't talk about it in this talk. And then I think subliminally or subconsciously, I got the idea of giving this talk from Gary Bernhardt, who's a PyCon attendee, who's given a wonderful talk called The Birth and Death of JavaScript. It's on YouTube, it's fantastic, and I took the California slide idea from him. And you should definitely go watch it. And I think that's all I have. So thank you very much for listening, and I'll take any questions. I'm really curious what questions there might be. I have no idea.
So this is Jason Chin from Pacific Biosciences. I'd like to
hey, Jason.
Get some of your comment about, like in biomedical research, there's a lot of data come from a lot of instrument provider. They generate a lot of data. How does the industrial's role in your kind of prediction that to be more open and sharing data while the industrial need to make profitable eventually?
Gosh. I guess I have two comments. So one is Cameron Nayland, I think it was, pointed out that back in the 80s and 90s, there were a lot of analytical chemistry instruments that automatically produced a lot of metadata with the data. And so when you posted the data, this is sort of not answering your question, but I thought I'd make the point anyway. So when you post the data, there was a lot of metadata that came with it in the bundle, not just a FASTQ file, or I don't actually know what PacBio produces with the traces, right? Not just that, but also the backstory, right? In order to run the sequencing experiment, you had to specify all that metadata. And I think that's a really good direction for instrument providers to go, because it means that you don't have to chase down the scientists later on. They just don't get their sequence if they don't give you that information up front. In terms of making a profit commercially from generating data sets, is that sort of what you were? How can you generate data sets and make them public while not sinking your?
I think there are two things, right? So we actually collaborate with a lot of people, and we really like to release the data, but actually not by our collaboration for that reason. So I think a perspective shift that I'm already starting to see.
is that data simply is uninteresting, right? So what I would like to see emerge is a situation where you post data sets as soon as they're available on something like Figshare or whatever, you get a DOI. When people use those data sets, they cite that DOI and you get recognition for that. The NSF has already made moves in the direction of recognizing data sets as valuable and on the BioSketch, and I think the NIH is following suit. So the perspective shift that needs to happen is for people to realize that the raw data is easy and uninteresting generally, and that the analysis and understanding of that data set in a particular context is what's interesting. And nobody else, there's a surfeit of data, nobody else is going to be, so one of my favorite things to say is the best way to sandbag your competitors is to release your data. Because now they have to analyze their data and your data. Right? And when the obstacle is data analysis, right? When the obstacle itself is data analysis, releasing more data doesn't help anybody. So I think I would expect there to be a perspective shift. You can already see this actually quite a bit. The biomedical folk are a decade behind. But, and then there's also privacy implications. And I don't know what to do about that. But I mean, apart from the privacy implications, data release I expect to become normal, frankly. You know?
So I'm sort of interested in why you're so down on benchmarks, because you have this thing of reproducible papers, and if you take a look at every independent algorithm, they of course do a little bit better than everybody else on their data sets. So why are you so against benchmarks in your predictions there, that they weren't useful?
I think you just answered your own question.
But if you're always validating against...
The algorithms always do better on their own data sets.
Right, but if the data sets that are the benchmarks are public and you're always testing against independent data sets.
Yeah. Okay, so.
And also evidence from other fields where benchmarks really move the state of those algorithms and fields forward by medicine.
So this is in partial response. I had a long conversation with the program manager at the Moore Foundation about metagenome assembly benchmarking. And what you've seen in the assembly field, with one major exception, which is the assemblathon, is that people publish a new assembler, and they take a data set that their assembler performs well in, and they publish those together. And that's the extent of the benchmarking. And then you get together and you say, gosh, I'm going to publish another paper where I take this public data set and I run a bunch of assemblers on it, and I get results, and I see that mine's better. Right? Nobody's surprised by this. In the end, that's the only way you can publish the damn paper, by saying that yours is better. I would argue that those, in the case of assembly, those incremental improvements are there's no ground truth validation, which is one problem. So it's always possible to get slightly better statistics by making bigger mistakes than the other assemblers, for example. And more to the point, I'm not really giving a very coherent answer. I'll probably have to write a blog post or something, an apology. But at the end of the day, a 10% improvement isn't interesting. 100 or 1,000% improvement is interesting. And benchmarking and engineering benchmarks reward the 10% improvement. And it's computer scientists are consumed with this idea that there's a well-defined problem, and the well-defined problem can be attacked and optimized, and then we're done. And we can move on to the next project. And I think it's holding bioinformatics back because most of the problems we have aren't well-defined. So I've sort of worked my way around to that statement. Benchmarks aren't useful because they're generally on well-defined problems, and most of our problems aren't well-defined.
But there's no reason that benchmarks have to be stable, right? Like you can get a better benchmark over.
Yes.
That's what Angel's trying to say is like
Sure.
a community benchmark that keeps improving.
Great. And when I see them, we'll have a different conversation.
Yeah. So like genome model, for example.
Yeah. So, and this is all deeply, there's a term I'm looking for. What's the term, Michael? There's a term I'm looking for where I am lying.
Bullshit?
No, not just that. That was the last hour. No, I am writing benchmark papers as we speak. Hypocritical. Thank you. Yeah. So, but the difference is, mine's better, right? The difference is that we are benchmarking entire protocols and real workflows on data, on cutting edge data, and we're not pretending that our results are correct. We're just saying this is what it is. And we're not trying to publish this as something exciting and new. It's sort of FYI, and they will be running benchmarks. Our Kamer counting paper, okay, so, well, what the hell? I have a couple of minutes to digress. So, so something I have not yet written up in blog posts. So we wrote a paper on our Kamer counter, and it's actually been accepted. It's the first time I've seen a Star Wars reference in a paper title. So there's that. It's these are not the Kamers you are looking for. Probabilistic Kamer counting with KHMR. And we wrote the paper, and there was jellyfish on KHMR, which is our Kamer counting system, and jellyfish, and we benchmarked jellyfish and Tallymer and I think one other. And the reviewers came back. I don't think either of the reviewers are here. The reviewers came back and said, well, you should just benchmark all the Kamer counters. It's like, okay. So, and actually, Nick Lohman said, that's crazy. You shouldn't have the author of a piece of software benchmark their software against other pieces of software because you know who's going to win. So anyway, digression aside, so we did the benchmarks, and because we'd written the paper in a 100% reproducible way, there's like 3 commands to go on a cloud computing system to go from the raw data to the final figures and published paper. All it took was about a day for my graduate student to add those new Kamer counters in and benchmark them and get them on plots. So that was pretty cool. And then a new Kamer counter got published. Like literally, we got like BF counter and whatever. We got the other 2 or 3 Kamer counters that were extant in the paper, and we're ready to send it back. And then another Kamer counter came out. And it was really interesting. This was K-analyze. And what was interesting is somebody in my lab sent around a note saying, "Hey, a new Kamer counter's out. I don't believe their paper." Okay. Because their paper said our Kamer counter is better than everybody else's jellyfish and Tallymer and KHMR and whatever. And my graduate student that evening, in response to that email, added it into our benchmarking suite, ran it, and showed that in fact it was much, 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 much slower. And the reason it was faster, ironically, and this was mentioned on Twitter, so I don't feel too bad saying it here, although this is all being recorded for posterity, anyway, was that they'd actually included the scripts that converted from other Kamer counters format to their format in the benchmarking. Right, so, okay. But it got published past the reviewers. I'm not actually sure who reviewed it, but we'll never know unless I file an FOIA thing. But what was cool about that, and what I think speaks to what you were saying, is that we actually are now setting this up as a running Kamer counting benchmark system, right? Hey, it'll take us a day to it'll take us an hour to add in a new Kamer counter. We'll run it on the cloud. We'll produce a bunch of HTML output tables. You can download the raw data, and if you want to argue with us, fine. We're running it on an Amazon system that you can buy, that you can rent. There's a GitHub. Everything's in GitHub. If you want to argue with us, tell us why we're wrong, right? And they won't be able to. So that's where I want things to go. But it's very rare to see that currently in bioinformatics because it's hard. Yeah, exactly. Yeah. I don't trust the dream challenges for one particular reason, but in general, it's a great idea. Yeah.
They're cool, so I think it's good.
Carol, yes.
So that's one of the old farts that you complained about. I actually have a slightly different experience, which is the old farts actually ones going, you know what, let's try and do something crazy. And it's actually the mid-career reviewers that try to stifle the notion of what is orthodox, beyond orthodoxy.
Okay.
So how
Yeah.
the worry is how do you kind of free up new career scientists?
Yeah. So actually, I'm not sure I disagree with you, although I'd bet that you're biased by who you drink with.
Yeah, I drink with you for a start.
Well, that's one of your many mistakes. But yeah. So I mean, you have to remember, I'm an assistant professor. So from my perspective, everybody is an old fart. So I don't really differentiate between sort of the mid-career and the senior people at my level. All I know is they spend a lot of time telling me that I should be paying more attention to my metrics and less attention to what it is I'm actually doing. Yeah, I have no answer. I don't know how to free up. I don't know how to free it up. I think one answer is, yeah, I have no idea. I guess the one comment I can make is I collaborate with a number of them, and the ones that don't get it are not going to have a career in 5 years, 10 years, because everybody is getting grants to generate data that they can neither analyze nor publish. And this is a real disaster in so many different ways. And only those people that are sort of willing to dive headfirst into this are making progress. So I have a great story. It's a keynote. I can be tangential, right? I have a great story. I spent 3 years working on what I think is probably the central achievement of the lab, which is this metagenomics work that we've been doing. And it was in collaboration with Jim Teege, who is one of these senior people in the field. And 2 years in, 3 years in, we felt we'd solved a bunch of problems. They ultimately got published, for whatever that's worth. And he went out to JGI to attend one of these community sequencing proposal meetings. And at JGI, and he was presenting our work. So JGI, for those of you that, I don't know how many of you are familiar with JGI, but the Joint Genome Institute takes in proposals to sequence stuff. They don't analyze it. They don't do anything particularly clever with it, but they'll generate the sequence and they'll give you the sequence along with a preliminary sort of basic stock analysis. And this turns out to be a disaster when nobody knows how to do the analysis, right? Because they generate a bunch of data and there's no follow-up funding for doing anything with the data. And that's actually how my career more or less got started, because Jim pitched this project to me and I said, sure, sounds like fun. And 5 years later, we finally got somewhere with it. So we went out there and he presented and he talked to everybody and discovered that nobody there knew what they were doing with respect to the data analysis. I think that's a fairly reasonable statement with environmental metagenomics. This was a couple years back too. But the funny and interesting part was he'd funded us to work on this. He'd collaborated with me for 2 or 3 years. And we got this phone call from JGI saying, I finally understand why you've been working on what you've been working on. Right? So he floated us for 3 years on the assumption that we probably weren't wrong to work on what we were working on. And I think that level of trust is what's required, right? Just to be like, well, they're not stupid, so probably they're not doing something completely wrong. Let's see how it works out. And I can tell you, from the administrative perspective, you never hear that from 80% of administrators that I talk to, 80% of career mentors at the university level would not have done that, would not have let me go that long. And I can tell you that because the other 80, Jim and a few others were saying, oh, keep on doing what you're doing, it's a big problem. And the administrators were all saying, you're crazy. So I don't have an answer, but let the crazy people do the crazy things. See what happens. All right, I think that's a good note to end on. Thank you very much.
Thank you, Titus.